Today, we actually have an excellent series of two guests. So first, we're going to have an old friend and longtime collaborator of ours, Scott Hansen. He's back for his second lecture, where he's going to tell us all about lipid sensing of mechanical force in the membrane and some really cutting edge new experiments he's been doing on how that interacts with Alzheimer's disease.
All right. Thank you, Keith. And thank you, Kim. So force is an important part of biology. My lab is interested in force. And in particular, at the molecular level, how does it work? And obviously, we've evolved to sense different forces. And we know that any type of mechanical force on a membrane can cause signaling in a cell. And this has been known for a long time, and we all experience this. For example, if we hit a funny bone, our hand actually feels pain or tingling sensation in the tips of the hand. And why would that be? Well, it turns out that it's not that your funny bone is actually sensing mechanical force. It turns out that that the nerve runs through the elbow and gets activated, and our brain perceives that as mechanical force. And so today we'll be talking about why that would be, and in particular, a general mechanism, underlying mechanism, and then how that would relate to ultrasound. And this picture here is showing that ultrasound can have the same properties as mechanical force, as just bumping your hand. So another place where we see a lot of mechanical force, and this will be important to today's lecture, is in the brain. And so your brain is constantly moving. And this is an animated, amplified picture of movements in the brain. But every time blood pushes into the brain, it changes the pressure, both in the arteries and then also in what's called the CSF, or the fluid that bathes nerves. And it can be pulsating. And under healthy conditions, this is always occurring because we always have blood flowing through our brain. Every time a nerve fires, there's also stretch. So the change of ions, if you have lots of salts going in or out of a cell, that causes stretching and shrinking, and those are all mechanical forces. So we're interested in these mechanical forces because ultrasound is a mechanical force. It's important to kind of have a perspective of how much mechanical force you would expect to see in the brain. And so here I've depicted kind of a range on the low and on the high end. So in venules in the brain, people have done studies where they watched how a fluorophore, how it moved through cerebral spinal fluid at the venules. And so this is where we kind of expect some of the lowest amounts of mechanical forces. And those go down to about 0.02 dynes per centimeter squared. And then you have arteries, and the cells lining an artery experience much, much higher amounts of force. They're somewhere in kind of max out around the 3 dynes per centimeter squared. And so there's, of course, lots of things all in between this 0.02 to 3 dynes. And this is considered homeostatic or healthy shear that you would, like that brain we just saw pulsating. These are the types of forces you would expect to be in that brain and going on all the time. But of course, there's disease, and there's also injury. And so we know that noxious forces can impinge on the brain. And we know they're noxious because we see neuronal death. There's hemorrhaging. So by the time the blood vessel just breaks and there's blood hemorrhaging into the brain, we know that that's noxious. And those are forces that nerves are not supposed to experience. And a lot of times we also can see edema, so extensive swelling. And this hasn't been characterized as much as it probably should be in disease states, these forces. But if you just take cells and you look how much force starts to kill a nerve, it's somewhere around the 10 dynes per centimeter squared. So anything getting above 10 dynes, we consider noxious, and then this is kind of your homeostatic range. So what are the mechanosensors? Obviously, the body has got to sense this stuff. And traditionally, ion channels have been the main focus of mechanosensation. But there's also enzymes and lots of other proteins in the body that respond to mechanical forces. I'll briefly mention a few mechanosensitive channels and kind of give you an idea of what that looks like, especially with some of the bacterial channels have been solved in multiple states and have really interesting conformational changes that are kind of self-explanatory visually. So we'll see a little bit of that. There's also a newer mechanism that my lab has been working on, which is sensitivity to the lipid membrane and enzymes coming together with their substrates and generating a mechanical signal. And there's also receptors in cell adhesion molecules, and I'm not going to go over that very much or at all today, but you can look some of that up if you want. So what is the molecular basis for force transduction? I'm starting off with this one because it's the one we'll talk about the most, or I'll show data on, and that's a disruption mechanism. And you can just imagine having two compartments, compartment 1 and compartment 2 in a membrane. And we know this now that there's compartments, and we'll describe those in a little bit more detail in the next slide. But for right now, I want you to just think for a second, if you have two compartments and they're separated by a distance, what happens when mechanical force impinges on the membrane or causes some type of mixing? We would expect that the components of those two different compartments would then mix. And would the body then, or would cells take advantage of that? And so this is the mechanism behind this, we think, is a thixotropy mechanism, and I'll describe that a little bit more later as well. Other very well characterized models for this, and some of these models are very difficult to actually test in vivo, so some of it's still theoretical, but one of them is a stretched induced thinning, and this is more popular for, say, bacteria, which stretch so thin that they're about to lyse. But this hypothesis is that there's hydrophobic mismatch and that the channels will actually thin, and then the stretch will cause them to splay out. And we'll see an example of that in a second. And the last example that is pretty well characterized, especially for eukaryotic mechanosensors, is stretching in the membrane. And so the membrane is elastic and it just stretches. And a radial force can pull, and then pull is thought to be able to pull a protein apart. Now, actually, how that happens in the membrane has not really been fully characterized. The proteins have been seen going through conformational changes, but how lipids would actually pull on the membrane, a lot of it is still at the theoretical level.
Scott,
So
are you ranking these in your
ultrasound.
order of like, are these ranked by which ones you think are real and which ones you think aren't? Stretch is like obviously
In
your
a
least
somewhat
favorite.
biased way, right? Because my last study is the first one. I ranked it in the order of what I thought was probably going on with focused ultrasound. Because focused ultrasound could cause some stretching. It could cause, it's difficult to understand how it would be radial, right? It's pulsed, and it's hard to understand how it'd be radial compared to, say, a swell of a cell. Some amount of stretch definitely could happen with focused ultrasound, but in thinking about a focused ultrasound, probably the most likely one would be this mixing mechanism. So that's what we'll focus on today. And so we would want a mechanism that one, we could directly test, and that would not necessarily be dependent on heat, and that could occur at very low energies. And speaking with some membrane bioengineers, or kind of the people who think about this is really a rheology problem. I want to talk about thixotropy. And what thixotropy is this concept. I'll just read it down here. It's a shear thinning concept, and it's a non-Newtonian fluid in which the fluid viscosity decreases with increasing shear stress. So let me explain what that might be. It's a non-Newtonian fluid, meaning that this isn't kinetic energy going into heat, right? This is non-Newtonian fluid movement. And you can I'll describe it and then I'll give you a comparison that you're probably more familiar with, which is water and ice. Lipids, and this is a depiction of lipids, they have long chain saturated carbons. And cholesterol is known in certain regions of the plasma membrane to pack with these lipids. And they do this by van der Waals interactions. And these are interactions that occur at very short distances, less than 5 nanometers. And they're very, very weak. But you need a lot of them. Or if you have a lot of them, then you get packing of these lipids, and the lipids actually become ordered and they become thicker. This is just a property of lipids with cholesterol, and it's a very important property of cholesterol in particular. There's also one other important concept here, and that's that a protein can be lipidated. What do I mean by that? Well, proteins can be modified after they've been synthesized. It's called a post-translational modification. And it's very common that a lipid will be covalently attached to that protein. And there's a particular lipid which is saturated called palmitate. And there's usually 2 of them that get put on in a very short amino acid sequence. And when there's 2 of these lipids and they're saturated, they replace a lipid in the membrane and they pack very nicely in these ordered domains. And here's just depicting that chain leading to the protein. And if the protein has this tag on it, it gets stuck in these ordered lipids. And so now we can finally get to our model of thixotropy. What happens is when you apply shear forces to anything that's ordered, it disrupts these really low energy interactions, and they move in 3-dimensional space such that they're not ordered very well anymore. And once they lose that order, it takes time for them to reorder or to come back. And so the temperature doesn't have to change. It's just that the movement of the molecules make it hard for those interactions to form. And so to give you an example of something you might conceptually be able to understand is the movement of water. So if you're below 32 degrees, water won't freeze. It's a pure liquid. Or if you're above 32 degrees. And when you get to 32 degrees, you can have ice in water. And if you add salt in high concentrations, or if you cause it to move, so if the water is flowing, it can't form that solid ice. And it's kind of the same concept. In the case of ice, it's a hydrogen bond. But in the case of lipids, it's a van der Waals interaction. But it's a way to actually disrupt or to change a state without actually changing the thermal temperature. So you can have water well below 32 degrees and it doesn't freeze. How would this be important to biology? Well, it turns out that these lipids that are ordered, they separate from the rest of the membrane. And so the membrane is kind of this patchwork of collections of different types of lipids. And we won't go into a lot of detail about these lipids other than to say that there are different types of them. So right now we know of at least 3 types of lipids. The GM1 are the cholesterol dependent ones that I just talked about. They're also where endocytosis takes place. So they're endocytic lipids. There's PIP2 lipids, and I won't talk a lot about that, but it's important to understand that there's these different compartments. And proteins can move between one compartment to the other. And when they move, they go from, say, really high concentration of cholesterol and no PIP2 to low concentrations of cholesterol and really high concentrations of PIP2. And these distances, we've been measuring them now are on the order of 40 nanometers. So they're about the distance of a synapse, and they're very close together. And so a molecule that moves from those 2 domains can signal very, very rapidly. So this is just to give you an idea of how that signaling might take place. And we call this cluster-associated protein activation. It goes from one cluster to another, and that activates a protein. So take, for example, this lipase that's called phospholipase D2. It's stuck in these GM1 domains based on its affinity from this 2 palmitates here. Well, when you apply mechanical force, through this thixotropy mechanism, or this proposed thixotropy mechanism, it disrupts the interaction with the palmitates, The PLD then no longer has affinity for palmitate, and it does have affinity for PIP2 because it has a PIP2 binding site. So let's go back. So it has a PIP2 binding site here, but it can't bind to PIP2 because it's bound to GM1. So as soon as this is disrupted, it now prefers to be with PIP2 and it moves. And as soon as it moves, it finds its substrate and it makes phosphatidic acid. So that was the first example that we published of how that cluster associated movement could activate an enzyme. So today we'll be talking about another enzyme that gets activated a slightly different way. In this case, there's a peptide that sits in the disordered region, which I'll describe later, is amyloid precursor protein. And its enzyme is in these GM1, these cholesterol-dependent clusters. And then when you add increasing amounts of cholesterol, the enzyme moves into this cluster and it finds, or not the enzyme, the substrate moves in this case. And then they are in the same compartment, and that presents the substrate to the enzyme and it cleaves the substrate. In a second, I'll show what that cleavage leads to. But it's important to realize here that the enzyme does not need to undergo any conformational change. This is just a movement alone is what causes the reaction.
Yeah, quick question. Would you say that all enzymes that are in the membrane have sort of an affinity in space? Or are there any enzymes that just move completely randomly? I guess any enzyme being there in the first place should have some sort of spatial order, or else it's just arbitrarily doing its thing, right?
In the membrane, if it's membrane associated, it's either palmitoylated or prenylated. So even like PKC and some of those, nobody's ever looked at it, but I'm pretty sure that they are spatially oriented, and this is probably going on, but we haven't studied that at all. In the cytosol, over the last 5 years, it's become very popular to study clustering or phase separation in even the cytosol. And so this concept of two things being compartmentalized by sequestration into clusters probably could be even happening in the cytosol. But in the membrane, if you're palmitoylated, it for sure. And there are lots of these, like all the DAG kinases, and there's lots of enzymes that are palmitoylated. Good question. So kind of the last technical thing before we move on to Alzheimer's proteins is these things are really hard to see. 40 nanometers, it's hard to imagine how small that is. And remember, this is the size of a synapse. You can't see this with a regular microscope. And so I'm not going to go into detail about this. You can look at this slide, and later, if you're interested in it, you can read about dSTORM imaging. But basically, it's a way to get higher resolution than a light microscope. Light is limited by the diffraction of light and the wavelength of light. And so you just can't see below 250 nanometers. And this is a technique where you measure multiple times the same fluorophore and you average where it is. So that if this is just a spatial representation, if you measure that fluorophore, say 20 times, and then you get a distribution, then you say, okay, on average, this is where it probably is. And it helps break that diffraction limited imaging barrier. And so everything we've done is with that super res imaging. Otherwise, you can't see it at all. And it does have some limitations. The other thing with this dSTORM imaging, how we determine whether things are in proximity is we do what's called two-color dSTORM. So we label two different things. Usually we label the lipid, like GM1, and then we label the protein. And we get these curves. And eventually we're all going to need to learn how to read pair correlation curves as dSTORM gets more and more popular. But how it works is you have what's called pair correlation, how much they're overlaid. And then you want to look at the pair correlation at really short distances. As you get further and further away, there's less and less correlation. And as you get closer and closer together, there should be more and more correlation if indeed the two molecules are in the same compartment. And so these are examples of what two different readouts would look like. This has very little pair correlation. This has a lot. And then we usually just use a bar graph at 5 nanometers. So just right here at the very closest place. So most of what I'll show you today is a bar graph coming from 5 nanometers, which is this pair correlation at this radius right next to the axis. Okay, so here's some data of what dSTORM looks like. We've labeled PIP2 and we've labeled PLD. And if you look in a confocal microscope, they overlay in yellow is where they're overlaid. And for the most part, it looks like PLD and PIP2 are together. But then if you do dSTORM imaging and you have super res capabilities, you can see that these are actually separated in almost all cases. If you go around and statistically add it up and you realize, no, PLD is not with PIP2, PLD is actually with GM1. And I'm not showing the GM1 data, but we can then use that 2-color dSTORM and label PIP2 and apply mechanical force, and you can see that move. I'm not going to show that today. I've shown that in the past. Rather, today we were going to want to talk about proteins that are involved in Alzheimer's. And we'll focus on 2 different techniques to look at Alzheimer's proteins that are pertinent to focus ultrasound. One is just the effects of fluid shear. We know that ultrasound causes shear forces in the brain. And we know that it's important that the fluid shear is that the cells have access to fluid shear. So we'll look at how that works, a molecular basis for that. And then we'll look directly in the brain at focus ultrasound, its effects on the brain tissue itself. And we'll compare a cell culture type environment where we're looking directly at shear to a focus ultrasound environment and see in super res imaging how those 2 things compare. Alzheimer's is caused by aggregation. Well, it correlates with aggregation of these amyloid beta plaques. And if any of you have ever had a relative that has had Alzheimer's disease, you probably have heard of these plaques that you get holes in the brain and you get these plaques. So where do these plaques come from? Well, they come from a protein called amyloid precursor protein, and it has to be cut by enzymes. And remember, we talked about those enzymes and how they would be controlled by cholesterol. Well, it turns out there's 2 really important enzymes. One of them is gamma secretase, and the other one's beta secretase. And if you get cut by both of these, you produce a little piece, a hydrophobic piece of protein from this APP that then makes these plaques. And there's 2 varieties. One's A beta 40 and the other one's A beta 42. And I'm not going to go into detail why that's important. The A beta 42 forms the plaques, but it's important to know that the enzymes make it. Remember, we said that the way you get ordering is with cholesterol. And this is a paper that my lab just published showing that we know in Alzheimer's disease that there is high cholesterol, or at least there's an enzyme that delivers cholesterol, and it's thought to lead to high cholesterol. And how this works is as you load the cholesterol into the membrane, and this is brain cholesterol, not the cholesterol that's in your blood. It's made by astrocytes, and it's specifically in the brain. And what happens is the APP moves into these lipid rafts, and then it gets exposed to both of these enzymes, gamma and beta secretase. So those are the examples I just gave you of an enzyme going into lipid domain. And the reason that happens is because this protein, APP, is palmitoylated. It has these palmitates. So it's tagged. It's tagged for these GM1 domains. And when you have high cholesterol, it goes in. So this is just depicting it another way. If you have APP sitting in the disordered region and you increase cholesterol, this APP goes in and it gets exposed to its substrate and it gets hydrolyzed. It makes the A beta, and then that forms the plaques that cause Alzheimer's disease. So what happens if we apply shear? Remember, we're really interested in fluid shears. The way we do this is we have an imaging chamber and we pump fluid across live cells. And we initiate shear, and then we add a fixative and it's fixed. And then we can image those with this super resolution image. So this is that APP protein. And we went from, remember our physiological range, right? From 0.02 dynes to 3 dynes is where we expect mechanical forces in the brain. And so you can see here, this is a control with no shear. And we have a hard time going less than 0.1 dynes. This is not very much force at all. And you can see with very little flow, very little shear flow, that APP comes out of these GM1 domains. And so now if we look at the membrane, this is what this would look like under all conditions of shear. The gamma secretase, which I'll show in a second where it goes, but at low shear, it stays in the GM1 domain and the APP comes out. So what happens? Well, when you have low shear, these two things are not in the same place, and then the A beta production goes down.
That effect is, I mean, it's really nice, especially with such a low amount of flow, but do you have to maintain it, or will these ordered domains come right back together if there's no shear force in the context of ultrasound or whatever
Well,
treatment?
we haven't done the experiment. Have we done the, I think we've done the experiment where we applied shear and then let it sit for 10 minutes and then measured again. That's the experiment you're asking, right? Like, can it just go back? Actually, I
Yeah, or
don't
how
think we've done
quickly it
that.
would go back even, like, is it a longer effect or is it like nanoseconds kind of thing?
That experiment needs to be done. We've talked about it, but I don't think we've done it yet. We've tried it with some other things, but I don't think we've tried it with APP. And it comes out so easily, it might take a while for it to go back in. So this is the data with gamma secretase. So now we've labeled GM1 domains and gamma secretase. Notice that these are at very different forces. So at 3 dynes was the maximum. Let me go back up. The maximum here was 3 dynes. And this is our lowest here, and it's still not out. It's barely starting to come out. And even at 10 dynes, we didn't get a whole lot. We had to go to 15 dynes, and then it robustly leaves. And so this is how we know that in this case, the gamma secretase is staying and the APP is leaving. What happens in a noxious environment? And I'm not going to show the data here because just for time, but we have done this with traumatic brain injury, and we do see this exact same thing. When you apply lots of mechanical force that's noxious, they both leave. And then now they're back together again, but they're in a different place. Now they're in thin membranes, which we know produces A beta 42. And so this mechanical force, high forces, is also activating. And this is kind of just a summary of how we think about this now. Shear forces are really important now for Alzheimer's. We know this now. And what we would expect is that you need, in a healthy brain, you need something between 0.1 and 3 dynes constantly going on in the brain. And so we think about this is, well, you need shear forces to just stay healthy. And if your brain stops pumping blood, then you're going to be unhealthy. Or if there's lots of things that could happen. You could have high brain cholesterol too. And then that would move this back in, and maybe you still have some forces, but you have lots of cholesterol that would cause Alzheimer's disease. We haven't studied this yet, but we think poor blood flow would cause the brain to then not perfuse as much. And if you don't perfuse as much, then you should start getting this clustering in the GM1 lipids. And on the opposite spectrum, if you have too much force, you move the opposite direction, but now you're going to produce A beta 42 instead of A beta 40. And anything involving hemorrhaging or edema, we think will shift over to. So there's the Goldilocks. You need a very specific amount of force, and we want that to be healthy. So what can we do if we have this case over here, for example, in Alzheimer's disease? We would want to know the amount of force that we could add that would cause it to come out, and not so much force that we would start causing problems. And so now we have a molecular tool set to look inside the brain during focus ultrasound and to know, are we helping it or hurting it, right? And so to do those experiments, we collaborated actually with Kim at Stanford, and they took a mouse brain and they did a similar experiment where you're perfusing with saline first while you're applying focus ultrasound, and then start perfusing a fixative under ultrasound stimulation so that the brain gets fixed in an ultrasound stimulated state. And I was really skeptical that this was going to work the first time. And we were very fortunate that it actually works. And this is the data from mice that Kim sent us. And this is APP with gamma secretase. Again, this is whole brain tissue from an animal. And we see the very same thing that we saw with shear forces on nerve cells in culture. And it dramatically releases APP from those GM1 domains. Importantly, we don't see any movement between gamma and GM1. So we don't see any differences with gamma secretase. So what does that mean? That means that there is at least some APP clustered with gamma secretase, even in a wild type animal, which we do see A beta 40 in a wild type animal. So it's not surprising there would be some there. And that when we apply this focused ultrasound, we now know that they are being separated and that you will produce less APP. And other people have done the experiment where they have applied focused ultrasound and they do see decreased products of amyloid precursor protein. So now what we have here is a molecular basis for that. And we can go and then use it to understand other proteins and also to fine tune exactly what we would want to accomplish a healthy brain. Probably exercising and doing the things we know keep healthy are probably healthy for the brain, but in cases where there's diseases and we can't control it, now we have a way to image and look to see that we're doing good instead of bad. So we're really excited about this data, and we think that this helps support this thixotropy mechanism of a palmitoylated protein being released from GM1 domains. So there's some suggested reading here. This talks about the control of a lot of what I talked about today, the background on that, of how cholesterol is controlling amyloid production in neurons. And then there's some other examples of where disruption of these lipid domains leads to activation of a protein. Before I wrap up, do you have any other specific questions or?
I have a question for you, Scott. You talked about basically what you need is a change of viscosity. And so my question is, do you really need shear forces, or what about if you just have pressure? Like you just put it in a pressure chamber, do you think it would have the same effect?
I'm not an expert on this, but I did talk to their collaborator at MIT. And apparently, yes, pressures are supposed to do the same thing. You actually don't need shear. So, and heat will do the same thing too, but heat is not necessary. So none of them are necessary, but they all, you just need to apply some type of stress, is my understanding. And I think in the case where people do see mechanical activation by stretch will also do this. So strain, bending a solid will also do this, and shear and pressure.
I was going to say, if any students have made it so far, it sounds like bad news for football players and good news for long-term ultrasound exposure, potentially.
Yes.
More to come from Scott in the future.